Okay, so our next speaker is Dominique Chung. Dominique started his career in Wisconsin and then Los Angeles, but in the field of microbiology and bacteriology. He moved to the University of Washington in Seattle and for some reason morphed into a biochemist, which is really a good thing because he's made major contributions in the field for many years. He's now a professor in the Department of Biochemistry and a member of BloodWorks Northwest Research Institute, previously known by other names, Puget Sound Blood Center. In the past, he's worked with Earle Davie. He was the first to clone the genes and cDNAs for fibrinogen, and that was followed by many more studies with Earle and on his own that have elucidated the structure and function of several coagulation proteins. And today he's presenting some data on how von Willebrand factor self-association is regulated. Dominique.
Thank you. Thanks for the invitation to speak here today. Von Willebrand factor is named after Eric von Willebrand. He was a physician in care of a kindred in the Åland Islands of Finland, and they have a bleeding disorder. So from those studies, it was identified that a plasma protein is involved, and a functional deficiency of that protein causes the bleeding disorder. So that protein was named after him. It is called von Willebrand factor, abbreviated nowadays as VWF. And the bleeding disorder was then called von Willebrand disease. So since then, a lot of studies have been carried out that characterizes how this protein works in normal hemostasis. That is, how it stops bleeding when there is a wound. At the same time, a lot of studies on thrombotic diseases started to implicate von Willebrand factor in the disease process. So one of the most interesting aspects of von Willebrand factor in thrombotic diseases was the disorder called thrombotic thrombocytopenic purpura, TTP. This is a disease that has been extensively covered in the last symposium, actually. And what it is that Moak in 1982 found that there was an ultra-large form of the von Willebrand factor implicated in the pathogenesis of the disease. And the characteristics is that, well, the characteristics are that they have massive microvascular thrombosis. And in the case of TTP, these platelet VWF thrombi occluded the vessels in the microcirculation, affecting multiple organs, and therefore it leads to multi-organ failure and, at that time, highly fatal. Nowadays, von Willebrand factor is also implicated in thrombotic diseases involving small vascular vessels less severe than TTP, in sepsis, in the hemolytic uremic syndrome, in cerebral malaria, and in sickle cell disease. Also, it has been implicated in genetic studies, epidemiological studies, that VWF may play a role in venous thrombosis, atherosclerosis, stroke, in other words, large vessel disease also. So, in the case of TTP, which we understand probably most, we want to take our lessons from what we understand in this context to see how we can study the other thrombotic diseases. In the case of TTP, von Willebrand factor and platelet thrombi are deposited in the microcirculation, occluding the vessels. And it consumed the platelet so that it shows up as extreme thrombocytopenia in the patient. And partial blocking of the vessels before they're completely blocked, circulation of red cells through partially blocked vessels causes the red cells to be shredded. So they are broken up, and so these patients have anemia. And in the circulation, you can actually observe the schistocytes. And these deposits occurred in multi-organs, and one of the organs that are most sensitive to this type of deposition is the kidney. So they have kidney failure most often. They have occlusions in the brain. They cause various degree of brain function, sometimes as severe as coma, sometimes it's as light as a headache. And from many studies by other investigators, it was shown that it is caused by the absence or functional deficiency of a plasma protease, a metalloprotease called ADAMTS13. We have since characterized this protein, we have cloned it, we have studied a lot of its characteristics, and it's shown that this VWF-specific proteases is responsible for cutting VWF in a very specific position and eliminated these depositions of the thrombi in the microcirculation. So in TTP patients, about 90% of them have circulating antibodies to ADAMTS13, and that's the reason why they are deficient, and that's why they accumulate these thrombi, and that's why they had organ damage, and they are in imminent, maybe imminent death. So von Willebrand factor is synthesized in two tissues, in megakaryocytes and endothelial cells. During the synthesis, it is dimerized in the ER into dimers, which are called protomers. And the protomers are then subsequently assembled into large multimers. And in this particular diagram, these multimers were displayed on a SDS agarose gel, which you can see the protomers about 500 kD, and they're linked together by disulfide bonds, and they reach upwards of 8 million, as shown in this band. And after assembly, part of it is constitutively secreted, and part of it is stored in the cells. In the megakaryocytes, which eventually mature into platelets, that they are in the alpha granules of the platelet. And in the case of endothelial cells, they are stored in storage granules, which are called Weibo-Palade bodies. Stored VWF are then subsequently secreted when the cells are activated by agonists. And so two publications appeared in 2002. One from the group of Ruggieri. They show that a functionally deficient von Willebrand factor, missing the A1 domain and therefore cannot bind platelets, when they were immobilized onto a surface, can regain the function of binding platelets if first normal von Willebrand factor was passed over it. So functionally, they implied that the defective one immobilized on the surface was able to bind to the wild type von Willebrand factor while it was passed over it and regain the function of binding platelets. So it is a functional complementation study that showed that VWF have the ability to bind to each other after they are secreted in solution into blood. In another study in the same year, which is carried out by Jose Lopez and his group in Houston, they showed that when endothelial cells are grown in flow chambers and stimulated by an agonist, in this case histamine, the endothelial cells secrete the von Willebrand factor from the Weibo-Palade bodies. And after they are secreted, portions of it remain bound to the endothelial surface. And on the endothelial surface, they start to bind to each other in a process which Ruggieri coined self-association, but it basically is a homopolymerization process, that more and more von Willebrand factors bind to itself and bind themselves in the direction of flow. So it is a flow-dependent or shear-dependent process. And in this case, after they are formed, when platelets are passed over the cells, platelets bind to these, von Willebrand factor assembled on the surface and gave rise to these beads on string-like appearance. These structures, the VWF in these structures that then at the time was called VWF strings, for short of anything to describe. These are massive structures. These structures, for example, in the diagram in the panel below, span across 3 microscope visual fields. When they're pieced together in this diagram, they reach a size of about 2 to 3 millimeters. And they're all capable of binding platelets. As you can see, they're all decorated with little dots. Those are platelets. So von Willebrand factor, when secreted from the endothelial cells, portions of it remain bound to the surface, self-associated in the flow to assume structures that are really huge. So this is the self-association observable, and this is the extent that the self-association can reach. In this experiment, in the same paper, at the time we purified ADAMTS13 from plasma while we are studying TTP, and we passed the ADAMTS13, which is VWF-specific protease. All of these strings were gone in 10 minutes. If we used the plasma from an acquired TTP patient means that they have acquired the ability to form antibodies to ADAMTS13. That's why they have TTP. So they have circulating antibodies specific to ADAMTS13. When we added the purified ADAMTS13 to that plasma, the antibody neutralized it, and when passed over these strings, did not touch the strings. However, when we use the plasma from a congenital TTP patient, these are patients who have mutations in the ADAMTS13 gene, therefore cannot make the enzyme. They don't have antibodies. When they are supplemented with purified ADAMTS13, they can clear all of these strings. So this shows the specificity of the cleavage after the strings formed complexes with the platelets, probably adhered to the microvasculature, could be removed if you now give him enough ADAMTS13. So these are the studies that we understand from the thrombi that was formed in this particular thrombotic disease, TTP. We know how it is cleared. We don't know how it's formed. We just know that they come out and they bind to each other under shear stress. So if we understand something about this formation process, we may have a better handle to look at some of the other thrombotic diseases in which VWF is involved, and also they have circulating near normal levels of ADAMTS13. So it is not the deficiency of ADAMTS13 which is causing thrombotic diseases in those cases, and we need to understand how the formation happens, or better, how it could be regulated. So that's what we started to do. We came into this problem from a different angle. So we were purifying von Willebrand factor. We were purifying von Willebrand factor and trying to study its behavior in how it interacts with ADAMTS13 and so on. And we were frustrated because when we purified the von Willebrand factor and put it in the test tube, we found that it just simply disappears. It disappears quite quickly. And the amount that we were lost to surface binding, in this case, was extensive. It was well known that proteins can bind to surfaces. This is the basis of how we do ELISA, how we do Western blots. But in cases when protein bind to surfaces, this should be saturable. It should be dependent upon the area. And when you bind enough protein to it, you stop binding. You can block it. You can do anything like that. But not with von Willebrand factor. When we purify the von Willebrand factor and put it in a test tube, static conditions, most of it would disappear overnight. So it must have been bound to the surface of the tube. So why do we lose so much von Willebrand factor in this case, more so than other proteins? We figure that, well, it must first bind to the surface, saturated the surface binding site, and now the von Willebrand factor is immobilized, was able to capture more von Willebrand factor from solution, forming a second layer or a second wave of binding. And the second wave bound molecules captured would also continue to propagate and capture more molecules onto it. So there would be initial interaction of protein VWF with the surface, followed by VWF interaction above it. So isn't that a form of self-association? So if we find out how we can interrupt that, maybe we can apply it to physiological conditions. So that's where we set off. We noticed that if we don't purify the von Willebrand factor, leave it in plasma, it doesn't disappear statically. So something in plasma must be interfering with this self-association process. So we want to figure out what is it in plasma that's doing it. So we fractionate plasma, added the fractions to purify von Willebrand factor in a test tube, wait overnight, next day, see whether it's still in solution. So we tried many things. Ultimately, what we ended up doing is that we realized that so we actually want to do a control experiment. What happens if we destroy all of the protein in plasma to see whether there is still anything still there? So we boiled the plasma for 10 minutes. We precipitated 98% of the proteins. We have still 2% of the protein that remains soluble afterwards. We tested those proteins. They can still protect von Willebrand factor from disappearing. So it is fairly easy. In that soluble fraction, you only have 3 major proteins. The 3 major proteins are apolipoprotein A1, alpha-1 ursomucoid, alpha-1 acid glycoprotein, other name is ursomucoid, and the third protein called transthyretin. And we can easily purchase these proteins pure and test them individually with purified von Willebrand factor to see which one works. And as it turned out that while the boiled plasma protected most of the von Willebrand factor staying in the fluid phase overnight, purified ApoA1, which is one of the major proteins found in the fraction, also able to do it, and perhaps do it better than the other 2 proteins, AAG and transthyretin. In the body, ApoA1 never exists as a free protein. It was always bound to lipid in the form of HDL, the good cholesterol. But this is the protein component of the HDL particles, which is also able to do to stabilize the VWF and prevent it from binding to the surface. We also found that when we dilute the surface with detergents, different detergents can dilute different things. We found that SDS, sodium dodecosulfate, was able to disrupt mostly protein-protein interaction, and in this case it's VWF association. They come out in 2% SDS. There's also a remaining fraction that bound to the surface of the tube much, much tighter than protein-protein interactions. They come out only if we use 4% chaps, which is another detergent. So using the different detergents, we can actually see that what was lost from solution, a portion of it is directly bound to the surface, but the majority of it is bound through protein-protein interaction, and in this case, VWF self-association. So this is all done in static case, and we know that HDL in our hands, in this assay, was able to stabilize it, prevent it from binding to the surface. So now what is happening with the real molecule in the real world? Shear stress, what happens in cells context? So we immediately do the experiment by exposing von Willebrand factor to shear stress and see what HDL is able to do. So in this experiment, we simply take purified or VWF in whole plasma, put it in a test tube, and start vortexing it at very high speed. And in this case, 2400 RPM on a vortexer for 90 minutes, or at room temperature, or at 37 degrees. With the purified protein, it disappears quickly. With purified 1-VWF, nothing left in the solution. With a biotinylated version of the VWF, this is VWF expressed by recombinant means, and it has a biotin attached to a unique position on the molecule, and we don't need to purify it. It's just stable in the culture medium, and when we start shearing it, it also disappeared. Most of it disappeared. We then go to plasma. This is citrated plasma, and we added EDTA to it because we want to inhibit ADAMTS13 from cutting the VWF in the first place, and just want to see what happened, shear stress is doing to the molecule in the context of a whole plasma with every other protein present. It also disappeared, most of it, and what was remaining are only the small molecules, the small multimers, which doesn't respond to shear stress as well as the large molecules. So this kind of makes sense. And then if we added HDL to plasma and do the same experiment, most of the VWF is stabilized and didn't go to the wall. This is the amount of HDL added, which is approximately 50% higher than what was already present in plasma. So these plasma, they all have a certain level of HDL and LDL and so on. And so what we're doing is that level is not sufficient to stabilize the VWF under this condition. We need additional amount of HDL which can stabilize it. So this is good. Shear stress is more physiological. So now we test the system in 3 biological systems to show how HDL may be interfering with the assembly of 1-Milgram factor. We go back to the flow chamber model where the endothelial cells are stimulated, in this case by phorbol esters. And without addition of HDL, we can see that a lot of strings formed, and when we pass platelets over them, we see these platelet decorator strings. We measure the length, quantified it, multiple fields, we get an average number of the amount of platelet decorator strings that appeared when these cells are stimulated. If we include the HDL during the stimulation process, we have much fewer platelet decorator strings. So this is consistent with observations already reported that HDL has a separate function, that by binding to scavenger receptors, it can downregulate or reduce 1-Milgram factor secretion. So less 1-Milgram factor secreted, of course, less string formation and less platelet binding. But if we stimulated the cells in the absence of HDL, let the 1-Milgram factor come out, and then add in HDL during the perfusion process where the assembly is supposedly to be happening, we also have a reduction in the number of platelet decorator strings. So this is consistent with the idea that it is interfering with the self-association process after they are secreted from the cells. So if we include HDL throughout the process, we have the fewest and shortest strings. So this is studying the 1-Milgram factor which is secreted immediately after stimulation. We are not studying the VWF in circulation yet. So this is one form of self-association that is happening and HDL is affecting. We then move to a microvessel system. This is a system where the endothelial cells are grown in the form of a channel, of a collagen channel, and they basically form a small blood vessel. In this case, we stimulated the cells so that they secrete the 1-Milgram factor, allow them to assemble into these strings. And in this particular case, these strings reach about 600 microns in length. They transverse, they bound to the channel and basically form transluminal fibers. They are really thick fibers. So now we perfuse through this system. After the fibers are formed, we perfuse through this system a biotinolated version of the VWF multimers. So now it's solution going through fibers. And we see that the biotinolated VWF bound to the fibers. So this is a different form of self-association by incorporating VWF in solution, grabbing it onto the fiber. In this way, the fibers are growing. They are thickening, or they could be lengthening. If we, on the other hand, before we perfuse the soluble VWF over it, if we pass HDL over these fibers, afterwards, if we perfuse the biotinolated soluble 1-millibranch factor, none of the soluble 1-millibranch factor bound, as shown in this panel. We go back and we look. There are 1-millibranch factor fibers formed in the first place, and we also saw the ApoA1, which is the protein in HDL. It itself had bound to the fibers. After it bound, the soluble 1-millibranch factor was unable to bind subsequently. So the perfusion of biotinolated VWF was unable to displace the bound ApoA1 also. So in this way, we are also studying a different form of self-association that is not only the ones that are coming out of the endothelial cells are able to self-associate, can grab things in circulation, things that have been exposed to plasma or to many other things. So this is a second form of self-association. We then go back and recently use a different system. This is a microfluidic device reported by a Diamond Group, and it basically contains a very narrow channel, 60 microns in width, and in the middle of the channel there's a post. The post is a square post, 30 microns in dimension, and the post extended from the ceiling to the floor of the channel. So when blood or fluid is forced through this channel, it has to go around the post. Where it's going around the post, the opening is much narrower. Therefore, it experiences accelerated flow. After it passed the post, the area again widens. Therefore, you have deceleration of flow. So in this system, we are studying the absence of endothelial VWF. We are only passing purified VWF in solution or VWF in plasma through the system and see what was happening. Just the effect of shear stress or elongation flow or disturbed flow on the process. In panel A, we simply circulated plasma through the system, and after 5-10 minutes, we can see these strings formed around the post in the direction of flow, and these are the VWF fibers that is formed. It goes fairly long down the channel. If we, on the other hand, added EDTA, which again inhibited the ADAMTS13 in the plasma to start with, we see a lot more VWF deposition around the post. One of our co-workers called this is the hair of Rapunzel. It is that massive, and it is only VWF. It doesn't have platelets in them. It almost occluded the entire width of the channel, 60 microns. This is the dimension of small blood vessels that VWF alone would be able to do. And when we added EDTA and HDL to the plasma, when you pass through the system, we have very, very few fibers formed, much, much less than Rapunzel. So in this system, we can also show that HDL has an effect on the assembly of these abnormal structures, massive structures in the vasculature. We extrapolated that. And so this is only with the VWF that's already in circulation, without participation of things that are newly secreted from endothelial cells. So in every case, we see HDL able to interfere with the self-association process. We then move to animals. We have ADAMTS13 deficient mouse, which do not spontaneously form, show the symptoms of TTP, unlike in humans where they would, unless these mice were challenged by Shiga toxin or something. In this particular model, the mouse was challenged by a massive dose of purified VWF. You have a sudden infusion. It causes, while it is circulating in the mouse system, it experiences shear, it binds platelet, so there is a transient thrombocytopenia. This is not a fatal model. They recover, but within 3 to 6 hours, there is a lot deep drop in the platelet count because it was bound up, forming complexes with the infused VWF, and these were deposited somewhere in the vasculature, so they are no longer in circulation. So transient thrombocytopenia is one of the characteristics of microangiopathy related to von Willebrand factor. So we are following that. And we want to see if we massively inject von Willebrand factor in this mouse together with HDL, what would happen? And it shows that the thrombocytopenia is significantly reduced, that the platelet count was much, much higher than just with von Willebrand factor alone. Not only that the platelets were not depleted, the injected VWF circulating level was much higher, also showing that they were not depleted or they did not bind to the vasculature. So it looked like a first approximation that HDL is having an effect not only in experimental system we see, but now in a whole animal, that it seems to have some effect. So in very recent studies, last week, we also studied what happened. Well, once we purify HDL, we also purify LDL, the good cholesterol, HDL, as well as the bad cholesterol, LDL. So we want to see what's LDL doing in this system. And so Alex St. John, who will be presenting something related to this system later on in today's talks, also participated in some of these studies. We show that if we circulate purified von Willebrand factor through the system, it can form these fibers. So this is the control. It forms. If we mix it with HDL, much less is formed. This we know, we confirm. If we put LDL into it instead of HDL, LDL facilitated the formation of these things too. So we have now a new function, component of plasma that would be affecting how VWF will be assembling and how hyperadhesive it gets and whether it is contributing to the thrombotic disorders would be something that we need to study. What we are doing by adding HDL is actually we are perturbing the existing HDL to LDL level. We can only add things, we cannot remove things at this time. So when we added HDL to the system, we are actually increasing the HDL effect, minimizing the LDL effect. So this is something that we are now focusing on heavily. So in summary, we now know that several things contribute to assembly of hyperadhesive, and perhaps maybe these are the things that is contributing to thrombotic disorders, even in the presence of ADAMTS13. Things that facilitate or stimulate secretion, therefore, is prothrombotic in this sense. So inflammation is known to stimulate endothelial cells, so they secrete a lot more VWF, so that it's more likely to assemble harmful structures. reactive oxygen species, for example, has been shown to modify VWF chemically and also modify the enzyme ADAMCS13 so that the cleavage process is inhibited or significantly retarded. So that also contributes in some way to the formation of these hyperadhesive VWF fibers. HDL is known to attenuate the secretion, so that may also play a role at that step. After the secretion, they assemble, and several things also facilitate the assembly. One, shear stress. So if we have abnormalities in the circulation, like constrictions or stenosis, where the flow of the blood through the vessel is disturbed through narrow regions and wider regions, those present locations of shear stress to the molecule, and that would also favor formation of these fibers. So site-specifically, it may be affected by shear stress. And also lipoproteins now, you may find they can also modulate this process. And ADAMCS13, of course, by cutting the VWF, would also eliminate or reduce the assembly. Platelet adhesion may be another step that this process could be regulated, although early studies with TTP, blocking platelet adhesion doesn't seem to be feasible. However, more GP1B specific blockade might be suitable in other circumstances. We don't know. We still have to study whether this is helpful or not. And then lastly, clearance of the von Willebrand factor platelet thrombi by ADAMCS13 is now being tried in some clinical trials that massive amounts of ADAMCS13 is infused into animal models, in large animal models, in pig models, and things like that in attempts to cleave off VWF platelet thrombi in thrombotic conditions. Lastly, I'd like to acknowledge my collaborators at the Research Institute, mostly by the support of Jose Lopez and other people, also from University of Washington. And I should also mention that the early studies where we are studying VWF disappearance was actually carried out while in the lab next to Earle.
Yeah, we'll just take one question because we're a little behind.
Sorry.
Hey, Dominique, have you or somebody else done a cryo-EM study of von Willebrand? Because you have a change in conformation, obviously, that allow you to go to this sort of a head-to-tail, not polymerization or fibril. It would be a very interesting thing to do.
Yes, those studies have been attempted way back in Houston, and I think they already had the data. We had tried also at the time to get Tamir interested in this, except that Tamir left. So we haven't done very much in that direction. But yes, cryo-EM probably show us a lot about the structure, the fine details of how self-associations is occurring, that kind of thing, how the strands are thickening, that type of thing. Yes.
Okay, I'm sure there's lots more questions. You can grab them. Okay, our next speaker is Alan Mast. Alan Mast is a senior investigator at the Blood Research Institute at Blood Center in Wisconsin. He received his Bachelor of Science in Biochemistry at the University of Illinois and then did his MD and PhD at Duke University. Then did some residency training in lab medicine at Washington University in St. Louis. So he sort of hit the high points in terms of coagulation and hemostasis thrombosis centers. So now he is at the Blood Center, as I said. He's got a well-funded basic science and clinical research programs, one iron metabolism and interest in the clinic, and on the other side, but in the lab, it's more in the tissue factor pathway, inhibitor pathways, so which is what he's going to talk about today. Alan.
right. First, I just want to thank you for the opportunity to speak here today, Ed. It's an honor to be here. And it's an honor to speak to Dr. Davie as well as to someone who can water ski like that. I'm still amazed. Okay, so my lab studies is focused primarily on this protein called tissue factor pathway inhibitor, which is called TFPI. Will be abbreviated as TFPI throughout this talk. It's an anticoagulant protein, so it keeps your blood from clotting. So you can look at it as bad for patients with hemophilia because it's keeping your blood from clotting when you want it to clot, but it's good for people that don't have hemophilia because it keeps you from getting blood clots when you don't want to that cause heart attack or stroke, etc. And one of the things our lab has recently done a lot of work on is this interaction that we found between TFPI and factor V, and that's going to be the focus of the talk today. So I want to introduce TFPI. This is the structure of TFPI. It's an alternatively spliced protein, and this here is TFPI alpha, and its structural consists of these three Kunitz-type serine protease inhibitor domains. So each one of these domains, in theory, could inhibit a serine protease. And what TFPI alpha does is this Kunitz domain can inhibit factor Xa directly, which is a blood clotting factor. And in the presence of factor Xa, it's a very effective inhibitor of 7A tissue factor catalytic complex that starts blood clotting. So TFPI is the main inhibitor in your blood of tissue factor initiated blood clotting. And this form of TFPI alpha is considered now to be a soluble protein, although occasionally it can be found on cell surfaces. Important for today's talk is this region right here, which is called the C-terminal basic region of TFPI, and we recognize that this has homology with part of the B domain of factor V, and trying to figure out what that was and how that relates to disease is going to be the focus of the presentation. There's also a second major isoform of TFPI that's called TFPI beta. It's produced by alternative splicing. It has these same two first Kunitz domains that inhibit 10A and 7A tissue factor, but it doesn't have this third Kunitz domain in factor V, so it's not going to do, it's going to have a distinct function from TFPI alpha in some ways. And instead of having those sections, it has this new or different region that promotes it encodes a GPI anchor attachment site that directly binds TFPI beta to the endothelium of the blood vessels. And so just briefly to tell you what I view, TFPI beta is what I like to call it a you didn't really mean that protein. And a you didn't really mean that protein is like if your blood vessel has some kind of inflammatory stimulus that makes tissue factor get expressed on that blood vessel and saying we want to make a clot, tissue factor will come and say I want to make a clot in this blood vessel, and the TFPI will say you didn't really mean that, you don't really want to clot off this vessel and have a heart attack, et cetera, or a DVT. And so it will dampen down initial procoagulant stimuli in that way. I want to talk more about this thing called platelet TFPI alpha. So TFPI alpha we've discovered is the only isoform expressed in platelets. And that's really another thing that started this whole project that I'm going to describe today is we were just looking at the expression of these two isoforms of TFPI in different tissues, and we found that TFPI beta was all over the endothelium and TFPI alpha was within platelets. And so this distinct tissue or cellular specific expression of the different isoforms intrigued us. An important thing about TFPI alpha that people often ask is it's not an alpha granule protein. I think it's just cytoplasmic. It is secreted. When platelets get activated, TFPI alpha will be secreted from the platelets, but when they're very activated, it will also be expressed on the surface. So this is an unactivated platelet here, and the green is TFPI, and if you look in there, you can see red, that's actin. And then if you highly activate these with thrombin and convulsin, you'll see the actin and TFPI will go to the surface of the platelet as well as being secreted. And so another initial question we had when we started studying TFPI alpha and platelets is we wanted to know, does it do anything, or is it just sort of there's a little bit of it in there, but it doesn't really have any physiologic function. So we did these transplants where we made mice that don't have platelet TFPI, and then we did a vascular injury model on them. And when we looked at them, we see here is a fibrin accumulation over time between the two, either platelet TFPI positive or platelet TFPI negative mice, and there's no difference. But when we looked at platelet accumulation at the site of the womb, when the platelets that lacked TFPI accumulated to much higher level, implying that it's much more likely to be an occlusive thrombus if you don't have platelet TFPI. So it's dampening platelet accumulation within the blood vessel. So we think it is physiologically relevant. And then one of our big discoveries, I think is a big discovery, is that we figured out that in addition to inhibiting 7A tissue factor, this TFPI alpha also inhibits prothrombinase, which is a combination of factor 10A, factor 5A, that can form on the platelet surface, and I'm going to talk about that, how that happens. So the biochemical mechanism for how TFPI alpha inhibits prothrombinase was we first came up with this clue where I pointed out earlier is there's this sequence called LIKTKRKRKK. All right, and this sequence is well conserved in both TFPI alpha and factor 5 in essentially all mammals. It's also in some fish and some birds, but not all of them. And so you just look at all of these, both TFPI and factor 5 have this sequence. All right, and no other proteins in the human genome have this sequence. So this is like, to me, this is like biochemist gold. You found something, and you got to figure out what it means. Okay, so this is what we did. And I want to go talk about some work that Rodney Kamari's lab had been doing. And this shows here the structure of factor 5, which is a heavy chain and a light chain, and it's separated by this big B domain, which doesn't really have any function that was well known other than just get clipped out so it could become active factor 5. And this right here is, this blue box stands for the basic region that has homology with TFPI. And over here on this end of the B domain is an acidic region. And what Mateen Boss and Rodney showed is that this blue box and this red box within the B domain of factor 5 interact with each other to main factor 5 as an inactive cofactor. So as long as the blue box and the red box are there, factor 5 doesn't get activated to factor 5A, and blood clotting doesn't happen. Okay, now if you take just trace amounts, nanomolar amounts of thrombin in nanoseconds supposedly, and expose it to factor 5, it will rapidly become activated, and thrombin will remove the entire B domain. So this was hard to understand. We were trying to think, why is this blue box homologous with factor 5 and TFPI if it just goes away when blood starts clotting? But I had the fortune of having Jeremy Wood come to my lab to be a postdoc, and he had just done his PhD work in Paula Tracy's lab in Vermont, and so he had studied factor 5 for a long time, and he came in and educated me, something I should have known, but he said there's other forms of factor 5A, there are fully active forms of 5A that retain this red box, and that these include when factor 5 is activated by factor 10A, or also if there's forms of factor 5 is in platelets, and there's active forms of factor 5A in the alpha granule platelets that have this structure that retain this. They retain the red acidic box, but they're missing this blue box. So what we hypothesized is that when these forms of factor 5A are used to assemble prothrombinase, which forms a fully active catalytic complex, TFPI will take advantage of its ability to interact with this red box. Since the blue box is gone, then endogenous factor 5 blue box is gone, so TFPI is now going to bind this, and it's going to allow it to inhibit prothrombinase. And so that's what happened, and this graph shows this data. This is prothrombinase activity on the y-axis and the increasing concentrations of TFPI on the x-axis. And here these open circles describe the reactions. We looked at how well TFPI could inhibit prothrombinase lacking the entire B domain, and it inhibited it with an IC50 of about 37.5 nanomolar. But the squares, we did it with factor 10A activated factor 5 that retained this red box. It inhibited with an IC50 of 0.9 nanomolar, so much 42-fold difference improvement, and we used 10A activated factor 5 rather than thrombin activated factor 5. And this circulating concentration of TFPI in plasma is about 1 nanomolar, so I think this brings it into physiological range as an inhibitor of prothrombinase. So now this is some new data I've never presented before, and so we wanted to understand more about what are the real biochemical interactions needed for prothrombinase inhibition of TFPI alpha. And I think this slide I just showed you showed that there's an acidic basic interaction between the red and blue box that's really needed for this interaction to occur. But we are also wondering, TFPI has a second Kunitz domain that's a direct inhibitor of 10A. Is that also needed for the interaction? And also if you look at the LIKTKRKRK region, it's very basic with all those basic amino acids, but the Li and the T amino acids are uncharged amino acids. They're not basic, but they're very conserved across all mammalian species, and we hypothesized that they would also have a role that's not charge-based. And so I work very closely with some investigators at Novo Nordisk, and we talked to them about this project, and they were able to very rapidly synthesize those forms of TFPI, missing different mutants of full-length TFPI. And so, and they did this in Chinese hamster ovary cells, and so they just gave us full-length TFPI alpha. In this case, we did the same experiment that I showed before, and here the IC50 is 2.1 nanomolar with this form of TFPI. But then we took active site altered TFPI alpha, where we mutated out the K2 active site so it no longer can inhibit 10A, and it becomes a much worse inhibitor of prothrombinase. So the K2 site on TFPI is important in addition to the acidic basic region for inhibition of prothrombinase by TFPI alpha. And then we also had this form of TFPI that instead of having Li and T, we made them ALA, ALA, and ALA, just changing them to alanine, and that is this purple line in here, and it also doesn't inhibit as well. So the Li and T amino acids are also very important for this interaction. And now we're in the process of trying to understand what these Li and T interactions interact with in factor 5A or factor 10A and make this prothrombinase inhibition happen. But it's more than just an acidic basic interaction. And so if you didn't understand all of that, I have this cool cartoon that's going to explain it to you in a second here. So here's factor 5. It's sitting on a membrane surface. It's bound to factor 10A, and this is acidic region, factor 5A, so it still has this red box. This B domain hasn't been totally cleaved. It's a fully active prothrombinase complex, and it's taking prothrombin and making thrombin. TFPI comes along, and we know the red box and the blue box can tightly bind to each other. 10A can bind to the active site of the Kunitz domain. I don't have in here the Li-KT interactions, but something's happening with Li and the KT to make this whole complex form. And now prothrombin's not being cleaved to thrombin because TFPI inhibited it. Now if you have thrombin come along, if you get further along in the clotting pathway and some thrombin's generated, it's going to come and rapidly remove that acidic region, and then we know once that happens, TFPI no longer inhibits prothrombinase and you get clot formation. So TFPI is only inhibiting prothrombinase very early in the clotting process. So just to summarize TFPI alpha inhibition of prothrombinase, this is exciting to me. This is the first described human inhibitor of prothrombinase. It's never been described a physiologic inhibitor of this complex once it formed, and now at least we have described something. This is isoform specific. It's only done by TFPI alpha. TFPI beta can't do this because it doesn't have the blue box. It requires K2, the Li-KT, and basic regions of TFPI alpha to do this inhibition. I think it ascribes physiological relevance to the homology of the basic regions of TFPI alpha and factor 5B domain. We originally identified and we're trying to figure out how that happened. And also importantly, I think it also identifies 10A activated factor 5, which happens before thrombin is generated, as a key point for physiological regulation of blood clotting pathway, which I don't think has really been focused on much in the past. So now I want to go and describe how we think this prothrombinase inhibition by TFPI alpha can affect bleeding and then clotting disorders. So first with bleeding disorders, there's this form of factor 5 called factor 5 East Texas that was characterized by Bjorn Dahlbach's laboratory, and it came out at the same time as we were presenting our mechanism for inhibition of prothrombinase. So it was a very exciting time. It's described in this paper down here and JCI in 2013. I definitely recommend if there's students or anyone interested in science to read this paper because it's a fascinating story. So factor 5 East Texas is a moderately severe bleeding disorder, and these patients produce through an alternative splicing event that's a mutation, an abnormal, somewhat abnormal mutation. They make a form of factor 5 that looks like this, that retains this acidic region, but it's missing the blue basic box. And this form of factor 5, which is really a form of factor 5A, is circulating in their blood, and it binds tightly to TFPI alpha endogenously. So these patients have this, and their plasma TFPI alpha level is increased about tenfold. And so they have this anticoagulant protein really high in their blood, and that's what's making them bleed. And I think it's because what they have is when you have the TFPI alpha factor V East Texas complex circulating in the blood, they have a circulating prothrombinase inhibitor. So anytime 10A is generated and tries to bind to factor Va, TFPI just inhibits it and they can't form a clot and they bleed. Okay, I also want to talk about, we do a lot of work with TFPI alpha and hemophilia, and this is just to briefly show the clotting pathway and why I think TFPI is important. Highlighted here, there's 3 main protease cofactor complexes in the clotting pathway. There's tissue factor 7A, there's 10A5A, which is prothrombinase, and then there's 8A9A, which are the hemophilia factors. And what I've shown you so far is that TFPI alpha inhibits both the 7A tissue factor and prothrombinase complexes. And in doing so, it can really alter the physiological activity of factors 8 and 9 that cause hemophilia. And because of this, there's several pharmaceutical companies that are now developing inhibitors of TFPI to try it as a treatment for hemophilia, and they're going into clinical trials. And as I said, I've been working with Novo Nordisk, just so as full disclosure, on developing their concizumab drug that was presented a little bit earlier today. And the exciting things about the possibility for this drug is that it will release early inhibition of both the extrinsic and the common pathways, and therefore restore thrombin generations in patients with hemophilia using a novel mechanism that's not replacement, but rather altering the blood clotting pathway. It will work equally well in patients with hemophilia A and hemophilia B, and it will also work in patients that have inhibitors to factor 8 or factor 9. It's been shown to be efficacious in several animal models in monkeys, mice, rabbits. TFPI can be targeted using several different types of molecules, and people have tried several different ones, and the most promising ones so far seem to be monoclonal antibodies. And the good thing about monoclonal antibodies is you can give them subcutaneously, and you can give weekly or less frequent dosing. So instead of having to do your intravenous therapies, I can envision, the fantasy is that little boys will be just getting a shot in their stomach once every week or once every other week and be able to go water ski or do whatever they want to do. And so just to sort of show where we hope, we will, the anti-TFPA antibody decreases hemophilia tail bleeding. So one thing that we did in collaboration with some scientists at Novo Nordisk is they just gave mice increasing amounts of anti-TFPI antibody in each one of these graphs, and then they looked at the 10A activity in plasma or the amount of blood loss in a tail clip assay. And what you can see is that the total plasma TFPI was inhibited at 2.5 mg per kilogram of antibody, but the amount of blood loss from the tail continued to decrease up to 10. And so we thought this could be happening because the anti-TFPI antibody is acting to block the platelet TFPI that's being released at the site of the wound. And so again, we did this fetal liver transplants into mice now with hemophilia to get either wild type TFPI or just no platelet TFPI. And this is this is the amount of bleeding from the tail of the mice that got TFPI in their platelets. And if you just took out platelet TFPI from the mice, it would restore their hemostasis and their tail bleeding. So these mice had TFPI all over their endothelium. They're just lacking the platelet TFPI. So to us, this was a very exciting result. And I've hypothesized from these data that a specific inhibitor of TFPI alpha and platelets may work for hemophilia treatment, and we're trying to do that. And that would have a big advantage because it wouldn't knock out all the TFPI all over the endothelial cells too. Okay, so I'll go to TFPI and factor V and clotting disorders, and this work was driven by this paper that was published by Dan Eitzman and Randy Westrick in 2002, and they looked at mice with TFPI heterozygosity, so they're missing one of the alleles of TFPI, and they had this mutation called factor V Leiden, which is a clotting factor, or a clotting mutation. And neither one of these mice individually have a severe thrombotic disorder, but when they were combined together, the mice died of severe thrombosis as they were born. And some of them, remember, this is factor V Leiden, which is half normal TFPI. They still have TFPI, but it's just half normal. And so just to tell you what factor V Leiden is, this is the most common hereditary thrombophilia in Caucasians. This ARG506 to Glen, and it makes them resistant in activation by activated protein C, which is another important anticoagulant protein. and they have increased risk of venous thrombosis. And for the first risk of VTE, you can see it goes up. If you're heterozygous, it's a 3 to 8-fold increased risk for VTE, or homozygous, it's 10 to 80. And what's very interesting is that plasma TFPI is decreased about 25% when women start to take oral contraceptives. And if you're a woman with factor V Leiden takes oral contraceptives, her risk for VTE goes up greatly. So at least it's mimicking the phenotype you see in the mice. So we think this could be physiologically relevant. And so our hypothesis was that TFPI alpha has decreased ability to inhibit prothrombinase containing factor V Leiden. And so we did thrombin generation experiments, and that started with 100 picomolar factor Xa, and this is normal factor V in these experiments, and then we had plasma from patients with factor V Leiden. It was approximately equal, but as we lowered the factor Xa to initiate these reactions, you can see that homozygous Leiden plasma clotted faster. The lag time is shorter here. And if you go down to 10 picomolar, you can see clots forming in the homozygous Leiden patients, but not in normal plasma. And the important part about these studies, when we added anti-TFPI antibody, that both Leiden and normal plasma clot exactly the same. So these differences in the initiation of clotting with dilute amount of 10A are totally dependent on TFPI. So now we think that factor V Leiden's procoagulant activity is mediated through two mechanisms. The first is the well-described factor V Leiden is resistant to degradation by APC, but we also think that factor V Leiden is resistant to inhibition by TFPI alpha, which affects the time for initiation of blood clot, or how sensitive a person is with factor V Leiden to initiating a blood clot when there's a small procoagulant stimulus. So to summarize, the factor TFPI alpha and factor V in bleeding. TFPI alpha is the only isoform in platelets. TFPI alpha binds to early forms of factor Va and inhibits prothrombinase, and early means 10A activated or platelet factor Va. Factor V East Texas bleeding disorder results from TFPI alpha factor V interactions that produce prothrombinase inhibition. And platelet TFPI alpha alters bleeding in hemophilia mice, and we think that maybe specific inhibition of platelet TFPI alpha may mitigate bleeding in hemophilia. For TFPI alpha and clotting, prothrombinase assembled with factor V Leiden is less susceptible to inhibition by TFPI alpha. So now there's two mechanisms for procoagulant activity of factor V Leiden, we think. Resistance to APC, which is a classically described mechanism, and reduced TFPI alpha dependent threshold for the activation of blood clotting. And we think this has physiological relevance, potentially both in mice, as demonstrated in the mice with factor V Leiden and TFPI haploinsufficiency, as well as women with factor V Leiden taking oral contraceptives. And that's all. Thank you very much.
Okay. Well, I'll start. It's interesting. You know, I work on complement, as you know, and there are all kinds of inhibitors. A major way in which complement is regulated is by destabilizing the membrane attack complex at various steps. And it's interesting you come up with this TFPI regulating prothrombinase, or disrupting it maybe, or regulating its assembly. Do you think that there are other circulating proteins that regulate the stability of the prothrombinase complex?
I like to be TFPI-centric and think how it's important it is. So, yeah, they haven't been described. You could hypothesize that any other protein that has a big basic region that would fit there might do it. But that's why I like the Li and the T amino acids are also really important for that interaction, I think, to really happen.
Alan, what proportion of TFPI do you think circulates with factor V?
So there is some TFPI. So they call that the form of factor V that is like the East Texas factor V short, and it's an alternatively spliced. So they have a mutation that makes an alternative splice that occurs rarely, and people without the mutation occur commonly. So, and it looks like all of us have some of that form of factor V East Texas in our blood, but I think it's about 1/100th or less. and that could be circulating bound to factor V. So the amount of factor V that's bound to TFPI is not very much, but I think about half of the TFPI is bound to factor V, because factor V has a much higher concentration than TFPI in the blood.
Is there a simple way of measuring TFPI? Because there used to be ELISAs that presumably measure both bits with no discrimination. Is there an assay one can use?
Yeah, we have we have assays in our lab. You can measure TFPI alpha well, and you can do total TFPI. It's hard to know what's the beta form specifically, because there have never been good antibodies to. There's just a little bit of TFPI beta that's not in TFPI alpha, and we haven't been able to make antibodies against that specifically. So you don't know if you're looking at degraded TFPI alpha or TFPI beta when you look at the shorter forms.
Thank you.
Just a quick question and follow-up. Is TFPI made in the endothelium or in the liver? And how variable is it to stimuli, like if it goes down with oral contraceptive exposure? How much variation in terms of inflammatory stimulus does it vary?
Right, so it's made in endothelial cells and megakaryocytes. It's not made in liver. And estrogens are one of the main things that really consistently have been shown to modulate TFPI plasma levels. There's other things that people have, you always see papers, inflammation affects this or that, but it hasn't been consistent. Estrogens are the one thing that make it really go down. Inflammation really makes tissue factor go up, and TFPI just sits there. That's why I always call it, you didn't really mean that protein. If there's a little bit of tissue factor, it'll dampen it, but if you have a big inflammatory event, tissue factor will just overwhelm the TFPI, and you'll get a clot.
TFPI beta and PNH clotting.
Yes, so that's been a long time hypothesis, and we've looked at that a little bit. I know George Brose looked at that extensively in his lab, and we haven't been able to prove that it's really related or not, mainly because it's hard to get the right samples from the patients with PNH. They're not willing to donate endothelial cells, and it's hard to get from the patients.
There's a question there.
Yeah, I was just wondering if you could comment on the release of TFPI alpha from platelets if they're found in the cytoplasm, and how they interact with factor V that's also released from platelets.
Yeah, so those are very good questions, and it's not super well understood. Thrombin will make TFPI be released from platelets readily, and if you put thrombin and convulsin, so you do something that stimulates the platelets enough to have a lot of phosphatidylserine on their surface, you'll see TFPI bound to the surface too. Is it bound to factor V or not as it comes out of the platelets? I'm hypothesizing it's not until they both get released, and then sometime after release it happens. But we're in a process of doing experiments that hopefully will answer that question. We don't know if inside the platelet, I don't think they are. Does that answer your question? I don't know if I
was just wondering if platelets need such a strong signal such as thrombin to release TFPI, wouldn't that also cleave the
Yeah, so that's
factor
an
V
issue.
region?
So how that all happens, you can also get TFPI released with collagen-related peptide. And so we think, that when does this really happen? I can't put it all together in my mind, because it's the same thing with TFPI alpha and platelets. It has to be you didn't really mean that protein, where you get a small stimulus where platelets get activated, and then TFPI says you don't really want all these platelets to get activated and make an intravascular clot. And then once thrombin is generated, TFPI doesn't inhibit prothrombinase. So I do not totally understand how all that happens yet, but I imagine it has to somehow.
Alan, thank you so much. I'm going to kill myself. Okay. All right. Our next speaker is Kay Toon, is a thrombosis research fellow at at the VGH, Vancouver General Hospital, supervised by Agnes Lee, who directs the thrombosis program. Kay completed her internal medicine and hematology training in Melbourne, Australia, and now is finishing dual training in clinical hematology and hematopathology at the Royal Australasian College of Physicians and Surgeons of Pathologists. She is going to really set the stage for a couple of other talks this afternoon on this particular topic with a challenging case of antiphospholipid antibody syndrome. Okay.
Thank you very much for the kind introduction. It's really my pleasure to be here and honor to present the interesting case of antiphospholipid antibody syndrome. You will also meet the patient, Mr. B, in the afternoon section, 3:15 session, when he shares his experience on living with antiphospholipid antibody syndrome, or APS. We also have Dr. Jacob Rand in the room, who is a world-leading expert on APS and his research. So I'd like to start with a brief background on antiphospholipid antibody syndrome, or APS, before start talking about the case itself. So what is APS? So simple term, it is an acquired clotting condition characterized by arterial clots such as stroke, or the venous clots such as DVT, deep vein thrombosis, or the pulmonary embolism PE, simply clot in the legs and clot in the lungs. The condition is associated with persistent presence of antiphospholipid antibodies. There are three types that we know of: lupus anticoagulant, anticardiolipin antibody, and anti-beta-2 glycoprotein antibodies. So some patients have a single antibodies, while the others have a combined. All three antibodies in the condition we call triple positive APS. Those triple positive APS patients are at the highest risk of clot or the thrombosis for both arterial and the venous thrombosis. Let's talk about so what do we do when patients with APS have the clot? We treat them with what we call anticoagulant therapy, or the blood thinners. So the most commonly used anticoagulant therapy is warfarin, which is vitamin K antagonist. And the other the type of blood thinner is the injectable form, low molecular weight heparin, which can be administered once or twice daily. It is usually used when the patient is intolerant to warfarin, or very unstable INR, which is a blood test to monitor warfarin activity in the system. So thirdly, we have newer types of oral anticoagulants, or the derived oral anticoagulants, or the DOACs. The evidence surrounding the use of DOACs in this setting is relatively weak at the moment. So I'm just going to, present about the case. We have patient Mr. Davie, 44 years old gentleman, who was a vice principal at the elementary school. He was first diagnosed with first episode of venous clots, DVT and the PE, back in 2002 in Edmonton, and subsequent investigations found that he has triple positive APS. So it means he has all three antibodies for the antiphospholipid antibody syndrome. So I'm just going to walk you through the treatment journey and the events for him. So he was diagnosed in 2002, and he took a year of warfarin therapy, and later it was switched to aspirin, or the ASA, 81 milligram once daily. So things were pretty quiet until 2010 when he experienced seizures. And MRI brain to investigate that showed the focus of seizures, and he was put on anti-seizures medications in this box, carbamazepine and clobazam. And subsequent follow-up brain MRI a year later showed that he had multiple areas of strokes. So at that time, you could see these stroke events occur while he was on aspirin. So another type of antiplatelets called clopidogrel and anticoagulant therapy, dabigatran, which is the DOAX, was added on as an anticoagulant therapy. So a couple months later in the year, he developed another episode of deep vein thrombosis DVT after a brief interruption of his antiplatelets and anticoagulant therapy for a minor surgical procedure. So it was thought that perhaps this episode is due to the drug interaction between dabigatran and one of these anti-seizures medications, carbamazepine, which could have resulted in the lower drug level of anticoagulant dabigatran. So after that episode, his anticoagulant therapy was switched to the injectable form, low molecular weight heparin. So now he is on what we call triple therapy, 2 antiplatelets with ASA, clopidogrel, and anticoagulant therapy with low molecular weight heparin. Then he started follow-up with VGH hematology in late 2011, the same year, after he moved to British Columbia. So when we first saw him, he's on triple therapy, and it was also noted that he tends to have a low platelet count. It goes as low as 70 or 80. The normal platelet count is somewhere around 150 to 400. So this is a condition called APS-related immune thrombocytopenia. Can be a frequent finding in people with APS. So now, due to the major concerns of bleeding on triple therapy and the low platelet count, and also a lack of hard evidence on the optimal therapy in this arterial thrombosis, with the discussion with neurologists, his antiplatelet therapy were tapered and reduced and eventually stopped. So aspirin was stopped first, and later Plavix. So around May 2012, he remained only on low molecular weight heparin. So there was no event until a year later in 2013. He experienced another episode of stroke while on low molecular weight heparin. So hence, aspirin was restarted. Things were pretty stable for about 2 years until late last year, where his family and colleagues noticed some changes in the memory. So the repeat MRI brain at that time showed that he had multiple new strokes, as well as some of his old strokes were getting bigger. So that occurs while he's on low molecular weight heparin and the aspirin. So in a desperate attempt to prevent another attacks of strokes, he was plasma exchange to bridge to the immunosuppressive therapy called rituximab. So rituximab is a monoclonal antibody which targets antibody-producing B cells in order to reduce all these antiphospholipid-related antibodies. So he finished his 4 doses of rituximab end of January, and about 2 months later, he started experiencing partial visual field loss, visual disturbances, which lasted about 20 minutes, what we call transient ischemic attacks. Quite frequently that we were afraid of crescendo TIAs, or he's going to have another major stroke. So he was then given another type of immunosuppressive therapy with intravenous cyclophosphamide, about 2 doses. He finished end of March that therapy. From there on, symptoms appear to have stabilized. Later on, he experiences some double vision or the diplopia. However, neurologists, we believe that it's probably the sequelae of the previous stroke rather than a new stroke. And luckily, the latest MRI in July showing stable appearance and no new strokes. So this is the combination of the journey he has gone through. So I'm just going to show you briefly about his antibody trending with the immunosuppressive therapy. As you can see, his baseline antibody levels with this anticardiolipin and beta-2 glycoprotein were pretty high. They both above 100. Normally the level is less than 20. With the plasma exchange, the level has gone down, and that fluctuates with other type of immunosuppressive therapy. The last level check in July showing some moderate levels of antiphospholipid antibodies. This is the serial MRI has been done. So this is how the normal MRI looks like. So in 2013, when he experienced a stroke, you could see all these white patches, the area of strokes. In 2015, late last year, when the new symptoms appear, you could see some of those old strokes, the areas were getting bigger, and they also found new areas of infarcts. So what we are doing now, what's happening, so we're just doing regular follow-up. Symptoms appear to have stabilized for the time being. We're just closely monitoring his clinical symptoms, biochemical markers, and also antibodies. He's currently remain on triple anti-thrombotic therapy with 2 antiplatelets, ASA, Plavix, and low molecular weight heparin, and also hydroxychloroquine, which is which is believed to have, which is immunomodulatory therapy used in lupus or SLE, believed to have some antithrombotic tendency. And we also, he's also on statin, anti-cholesterol, and also a couple of blood pressure tablets to try to optimize his other cardiovascular risk factors to prevent another stroke. I'd like to thank the patient, Mr. B, for allowing us to present his case and discuss in this meeting, and also the organizers for the symposium, and Dr. Agnes Lee for giving me an opportunity to present, and also Dr. Paul Jensen, patient hematologist, for his advice on the timelines of events and also the slides. And lastly, thank you very much for listening.
Any questions? Comments? Yeah, none. No comments. Jake, come on. There you go. Twist it out of him.
Well, thank you. I think that, well, to a degree, just a little bit, I'm in awe that the patient is here as well as the presentation. And this is really, it's what you're describing is a testament to the significant progress that's been made in a condition that for which the mortality was greater than 50%. I think that it also exemplifies the importance of making additional progress here. And the last thing that I just want to say is that on a practical level, an additional modality that's been tried in patients who've been refractory to the current treatment, and thank goodness Mr. B is doing fine apparently with the present treatment, has been the potential of using complement inhibitors, and I'll touch on that during my talk as well. But again, thank you, Mr. B and family, for coming, and thank you for presenting the case.
Thank you so much for coming.
Thanks, Kate. Okay, I need the shotgun presenters. All you who are presenting, 20-second presentations. Quick, 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 because we want to eat lunch. I don't see you moving. Come on, let's go. Come on, you're young. Hustle, hustle. All right, so this is going to be lickety-split. These are about 15, 20-second presentations related to posters that are up there. Most, but not everybody, are presenting. So one after the other because we're hungry. Okay, go. Brian, you can start. Go.
Okay,
Yeah,
cool. Hi, my name is Brian Lin, a PhD student in the Preisel Lab, working towards broad-spectrum antiviral strategy. And in my poster, I'll be showing you an electron microscopy technique that developing to identify host constituents that we believe or hypothesize are found universally on several important viruses. As models, we're using purified dengue virus as well as herpes simplex virus. However, this idea can be applied to other important bloodborne viruses such as hepatitis C, HIV, Ebola, Zika virus, and West Nile virus, all of which have an envelope and likely derive the same host protein. And I will also show you that this host protein is vital for starting the host clotting system and ultimately leading to pathology and infection. If you're interested to learn more about viruses, envelope viruses, and there are links to blood pathology, visit me at poster number 6. Thank you.
Hi, my name's Stephanie from the Castrop Lab at UBC. So platelets are natural delivery vehicles delivering small molecules, proteins, and nucleic acids to sites of damage within the vasculature. Our lab hypothesized that we could use platelets to deliver nucleic acid-based therapeutics to sites of damage by using lipid nanoparticles to transfect platelets and having them release this RNA back into this blood system. We've shown that we can deliver RNA with these lipid nanoparticles, and they cause minimal platelet activation and aggregation, and the RNA is also released in microparticles upon platelet activation. So if you want to hear more, come check out my poster number 30. Thanks.
Hello everyone, my name is Ahmed Arbain and I'm from Dr. Divine Lab. My poster today is about an in vitro simulation of an in vivo situation where I see the effect of Mirasol treatment on the hemostatic functionality of whole blood treated units. Please visit me at poster number 2. Thank you.
Hi, my name is Cecilia. I'm a postdoc in Dr. Q Kim's lab. My research focuses on the interaction between platelets and implant biomaterials. An important goal of tissue engineering is to achieve optimum integration of host-bound tissue with implanted biomaterials such as dental implants. It is well known that implant surface has the ability to determine the quality of bone-implant interface as it influences the fundamental cell functions involved in the development of the interface. Platelets is one of the first cell types to contact an implant surface after surgical placement. It contributes cytokines and growth factors which are crucial to wound healing and also integration. However, there's very little known about how surface topography affects the behavior of platelets. We conduct our study with a view to improving our understanding of how surface topography regulates platelet behavior. If you're interested, please come to my poster number 7. Thank you.
Hi everyone, my name is Anoli De Silva and I'm a master's student from the Kim Lab. And we're studying the molecular determinants of platelet apoptosis. So platelets, as we know, have a lifespan of around 10 days, and there's different mechanisms that can regulate this, including apoptosis. However, the exact biochemical pathways and the signaling mechanisms are not clearly defined, and so we set out to determine whether the role of intracellular calcium released from the dense tubular system can induce apoptosis in platelets. After inducing apoptosis using a SERCA inhibitor known as thapsigargin, we found that platelets do display the markers of apoptosis. And so this will be one of the first studies to show that the release of intracellular calcium can induce apoptosis in platelets as well. So if you're interested in learning more about this topic, you can come visit me at poster number 11. Thank you.
Okay. Hi, I'm Hura, and I'm a postdoc at Ed Conway's lab, and I'm studying the effect of thrombomodulin on a laser-induced model of age-related macular degeneration in the mice eye. So if you are interested, just come and meet me at my poster number 14.
Hi, my name is Vivian from Dr. Christian Kastrup's lab here at UBC. So as you guys know, platelets are amazing cells that are involved in many physiological and pathological processes and represent attractive disease targets that we can treat. However, manipulating the genetic and proteomic content of platelets is not a trivial matter. In a proof of concept experiment, we have showed that we can load platelets with nucleic acids, with proteins, as well as small molecules to actually transcribe RNA inside of platelets. So I'll be at poster 35, and I'd love to chat with you about some of the potential applications of our technology. Thank you.
Hi, everyone. I'm Parker Jobin from the Overall Lab, and I study how intracellular cytosolic proteins can actually get released into the extracellular environment. Being a protease lab, we found some candidate substrates that were only expected to be in the cytosol. However, we found that when we treated them with interferon gamma, macrophage could actually secrete this tryptophaneol tRNA synthetase to get out there, and once it's out there, it can actually bind back to toll-like receptors and induce an inflammatory response. So if you want to find out more, come to station 23 at my poster.
Hello, everybody. I'm Anthony from the Cote Lab. And I'm presenting a poster today about mtDNA in platelets. Now, mtDNA in whole blood has been increasingly commonly used as a marker for mitochondrial dysfunction. On my poster, you'll see lots of numbers, correlations, multivariable models, which are very boring. If you come to my poster, I'd love to talk to you about the implications behind those numbers, specifically the intersectionality between mitochondrial dysfunction and aging. I'll welcome any arguments about the numbers or discussions about the concepts, or vice versa, at poster number 4.
Hi, I'm Simon. I'm from Dieter Brahm's lab. Our lab is interested in studying an enzyme called cathepsin K, which is highly involved in bone remodeling and has recently become a target for treatment for osteoporosis. One of these drugs is odanicotib, which has made it through phase 3 clinical trials before being abandoned. This drug is actually highly selective for the human enzyme over the mouse enzyme, and through crystallographic approaches, we determined the structure of the human and the mouse enzyme and identified 2 structural elements which mediate the inhibitory effect on the mouse enzyme. And through mutation analysis of the mouse enzyme, we were able to create a mouse enzyme which is receptive to odanicotib. Thank you. And if you're interested to learn more, my poster number is 29.
Hi, my name is Karen. I'm from the Dr. Christian Kastrup's lab. And so, as we all know, bleeding is bad, and we want to develop a new method of treating bleeding. So for this purpose, we developed a fibrin-mimicking polymer. It's a synthetic material, and it can be blended in blood to increase clot adhesion even under fibrin-poor and fibrinolytic conditions. So if you want to learn more, please visit my poster. It's poster number 19. Thank you.
Hi, my name is Sarah, and I'm a shared PhD between the Netherlands and UBC in chemistry. And I'm working with Chris Orvik on the synthesis of new radiopharmaceuticals. But right now I'm working with Ed Pricedale in a collaboration where we try to develop a new theranostic agent which can image blood clots. And we're doing this by having a bifunctional chelator which can bind to gallium-68, which, as you might know, can be used in PET imaging. And we also use a biological vector, which is in our case factor 10A. So right now I can present on my poster 27 a lot of beautiful chemistry. So if you're interested in that, please join me at my poster 27. Thanks.
Hi everybody, my name is Osama and I'm a PhD candidate at Dr. Kizakaduktu and Dr. Brooks' lab. My poster for today will illustrate the first couple of steps we've made in developing a novel liver-targeting iron chelating system for the treatment of transfusion-induced iron overload. I'd be happy to entertain you at my poster number 34. Thank you.
Hello, my name is David Rattray. I'm from the Foster Lab. My research focus is on the spread of salmonella through the blood during some systemic infections. My poster today is looking at the suppression of surface expression of MHC2 complexes and how that affects the adaptive immune system. Thank you.
All right. Hi, my name is Eva, and I'm poster number 12. So germline genetic variants can influence a patient's response to chemotherapy. In the clinic, tumor biopsies are collected without match normals and are formalin fixed, paraffin embedded. Formalin causes sequence artifacts in DNA. I showed that without match normal, we can accurately detect clinically relevant germline variants in formalin-fixed, paraffin-embedded tumors. If you want to know more about my research, come to poster number 12.
Hi, my name is Abhinav, and I'm also a student in the Kote lab. Disclaimer, we do not work on factor 8, factor 10, or 1 milligram factors. Our lab investigates the effects of HIV and antiretroviral therapy exposure on biomarkers of aging, such as telomere length and mitochondrial DNA damage. For today's poster session, I will be presenting the results of a study where I investigated how the number of antiretroviral drugs taken by moms during pregnancy affects their infant's mitochondrial DNA content. If you're interested to learn more, please come by my poster. It's poster number 1. Thank you.
Hello, I'm Chahat Singh, and I'm from Dr. Wilfred Jeffries' lab at the MSL. And I'm working on looking at the vascular pathology of Alzheimer's disease. And it's seen that angiogenesis is upregulated in Alzheimer's patients, and there's a lot of, clotting and inflammation, and all of these pathways are involved, and that upregulates angiogenesis. So I'm trying to look at using anti-cancer therapeutics to fight angiogenesis and treat Alzheimer's. So I'm at poster number 37. I would love to have inputs from all the experts on vasculature out here. Thank you.
Hello, my name is Arya, and I'm in the Healthy Art program at St. Paul's Hospital. Familiar hypercholesterolemia, or FH, is one of the most common genetic diseases characterized by lifelong elevation of LDL. These untreated individuals have a 20-fold increased risk of premature cardiovascular risk. So the BCFH registry was established in 2012 to diagnose, educate, and treat these individuals from the silent killer. Come to poster number 5 to find out more.
Hello everyone, my name is Piyush. I am a postdoc at Conway's lab. So today my poster is on the role of complement in venous thrombosis with objective to delineate the molecular link between complement and coagulation using genetically modified mice. And our preliminary data shows that complement C3 deficiency enhance resolution of clot after dalteparin treatment, which is low molecular weight heparin. So please stop by at poster number 26 for more information. Thank you.
So that wraps up this morning. Thank you to all the speakers and all those younger people who just gave little talks. So thanks to everybody.